Hello and welcome to this webinar jointly organized by the Journal of Ayurveda and Integrative Medicine and Patanjali Research Institute, Haridwar. I, Sanchita Sangre, welcome all our speakers and participants for this event. Technological advancements have enabled research and development in modern biology. Currently, these advancements are shaping Ayurveda practice and research. Keeping that in mind, we arranged this webinar on advanced technology for Ayurveda research. The upcoming JIM special issue on Ayurveda and technology calls for articles related to advanced technologies capable of enhancing Ayurveda education, practice, and research. I'm happy to introduce Dr. Prashant Gupta, who will be the moderator for the webinar. He's a Vaidya scientist with expertise in Bal Rog. Currently, he works as an associate professor in Department of Bal Rog in All India Institute of Ayurveda. He is a DHR Winter Fellow and an INSA Visiting Scientist Fellow. He has recently established the Ayur Informatics Lab at All India Institute of Ayurveda for in silico research and training of Ayurveda researchers. Over to you, Prashant, sir.
Thank you very much, Sanchita. So, before starting this seminar or webinar, I would like to thank Savitribai Phule team and team Patanjali for initiating such a beautiful initiative. So this is the first webinar in a series of technology and Ayurveda. So organizers conceived the idea of conducting such webinar and understanding the need of the hour. So the main of this webinar is to inculcate the technology in Ayurveda. What is good already is going on or underway in Ayurveda. The third one is what are the newer meadows or the interfaces of Ayurveda and technology. So as a potential contributor to Ayurveda, expectations from SSPU and Patanjali and All Indian Institute of Ayurveda are high. So here it is our first step in the said aim. I personally and on behalf of this organizer team, I welcome each and every fellow. So we start with our first speaker. Am I audible? Okay. So our first speaker is, I would like to introduce Dr. Manali Ji Joshi. She is a master in biotechnology from IIT Bombay and followed by PhD in computational biochemistry. Dr. Manali Joshi is instrumental in research of molecular modeling and simulations, and her research interest includes computational approach of drug design and understanding the structure, function, inhibition paradigm of protein. So I request Dr. Manali Ji Joshi for a talk over computational approaches to enable research in Ayurveda. Over to Madam Manali Joshi.
Thank you, organizers and Dr. Gupta, for this kind introduction and opportunity. Am I audible?
Yes, madam, you are audible. Please continue.
So let me try to share my screen. Okay, so let's begin. So what I'll try to do in the next 20 minutes or so that have been allotted to me is I will try to give an overview of the different kind of computational approaches that can be used to enable research in Ayurveda. So let's first talk about the computational revolution. So ever since computation or computers were, let's say, invented, it actually revolutionized all areas of life, including science, including biology. And since all the members here either are related to Ayurveda or life science, in some way or the other will agree with me that computation is now, or computers are now, an integral part of our life. So how actually did they create this revolution? So I would say in three major areas. One, using computers or using IT, what we have been able to do wonderfully is to organize, store, manage data which makes it very easy to retrieve it when we want. So imagine if computers were not there and I wanted some protein sequence or some information in a pre-computer era. We would have to go to the library, hunt up the journal, open those dusty books, read something, interpret. So it was so time taking and difficult. And now you can just sit and the click of a button, you have so much knowledge, or rather not knowledge, but information available at your fingertips, which then you can convert to knowledge, right? So one of the biggest revolutions that computers have done for us in terms of life science, and which hence I will also translate and impress upon you, it has the potential to do that for Ayurveda as well, is organizing data in a very user-friendly manner, which will make it accessible to a lot of people. And we'll take some examples as we go along the way. The next area, okay, where computers have played a very important role is once you have a lot of data, you need some tools or algorithms to analyze or interpret that data. So pre-computers, it wasn't even possible to look at so much data together. But now with smart algorithms, bigger storage, lot of memory, we can actually handle a lot of data with a lot of statistics. We are able to actually do data analysis and interpretation. And the last part is actually dissemination and visualization. So once you have the data, actually presenting it in a way which will be user-friendly, easy to understand, and then ultimately making some knowledge out of that data by analyzing and interpreting. As I'm going ahead, I request you not to annotate the screen, because if somebody does that, I'm not able to erase it. So it's a request to the participants to please not annotate. If you have a question, please feel free. You can interrupt and we can perhaps take some questions. So whoever has done this, I request you to please, if you can, get rid of that. Okay. So
we request participants to please follow the instructions.
okay, so as we know, Ayurveda is actually the science of life, let's just say, right? It's knowledge about life. So to call Ayurveda just as medicine or diet or all the points that are in this slide is absolutely not correct. But one of the lowest hanging fruit, as we would say, which is most approachable to people who are trained like us, is to start immediately looking at herbal medicine. So where are we coming at today? So today, Western medicine is very, very well accepted because it follows the rules as per new science, how science has evolved. So the way technology evolved, Western medicine evolved, and we all practice and accept it. Now, many traditional medicine systems, including Ayurveda, for that matter, other systems in other parts of the world, they were something that were discovered, developed, and written many, many centuries ago. So to understand or bring them up to today's speed in today's context, we need to give it relevance by using today's tools and techniques. So what I will try to do is, instead of looking at all of them, I will focus on this one area of Ayurveda. And once again, like I said, herbal medicine is not equal to Ayurveda. So I am not impressing this upon you at all. Ayurveda is, in fact, a way of life. It's just that my talk will be focused on this and how we can better understand this, because this is the lowest hanging fruit. And in fact, I would like to say that a lot of people have already done a lot of research, let's say, for instance, on meditation, where I have read or gone over some research papers where people have taken EEGs of the brain and found that when you actually go into deep meditation, what are the things it does to your body, the alpha waves that are produced in your brain, how it induces calmness. And in fact, even about yoga, there are so many scientific studies done. So today's talk, or what I will be discussing more, will be focused on herbal medicines. And let's see how computation can play an important role in this area. But there are many, many areas, and maybe at the end of the time allotted to me, I can offer a few suggestions where we can have a handshake and evolve in all of these different dimensions. So this is where I will be focusing my talk on. So like I said, databases are very, very important. And I'm sure the speaker after me, Dr. Saket Ram, who represents the ministry, will actually tell you not just this traditional knowledge digital library, but perhaps about many other initiatives that the ministry has taken to digitize the beautiful records that we have regarding Ayurveda, or perhaps other traditional knowledge systems also. So this is just one example of a database that is very, very user friendly. So if you go and read, and I'm sure quite a lot of you might be already knowing about traditional knowledge digital library, that the people are also very much involved in looking out for who else is trying to patent what we already have as part of our traditional knowledge. So such compilations which have been done or can be done will play a very, very important role in taking Ayurveda to the next step. So let's talk a bit more about herbal medicines now. So unlike Western medicine, which we know evolved as, let's say, a single chemical which works on a single target and a single disease, I think most of us will be aware that the medicines or the therapies that are provided by our Ayurveda text work a little bit differently. Okay, so I'll just take a couple of examples. For instance, haritaki fruit is one of the, let's say, one medicine that is given. And it's not just it's given, but it's the solvent also, or the carrier also, which can change. So for instance, haritaki, when it is given with ghee, it may be prescribed by a physician for some neurodegenerative disorders. Haritaki, perhaps when given with salt, can be prescribed for digestive disorders. When given with water, can be prescribed for inflammatory disorders. Now, this is unheard of in Western medicine. It's not like you're prescribed aspirin with water for X or aspirin with oil for Y, right? So this conceptually, this is very, very different. And there is no science right now to explain this. Okay, so there is so much to be done to understand. And this is something we have just started looking at, that one fruit, okay, how it can be used, or how the ingredients in that fruit can be used in a plethora of different disorders that are described. So we'll discuss in a moment some methods that are evolved or evolving, which are very popular to try and understand this. On the other hand, so this is very unlike Western medicine, right, which we don't understand is that how very well yet is how one can be given with many different carriers for many, many different diseases, right? So how do we understand this better? Now, another complicating layer is most of the times the formulation is not just one, but many. For instance, Triphala. I'm sure most of you also have heard or know about this. So Triphala is actually 3 ingredients which can be given with so many different solvents, and you can see however how many diseases are listed, right? So this is very, very unlike Western medicine, and we really need very robust scientific systems to understand this in a much better way. So many people are practicing this. We agree that they are beneficial, right? But what now we need to do is, in a scientific and rational way, try to explain this with evidence. Okay. Now, before we go to how people are doing it right now, let's try to understand one main component. So when we talk about herbal medicine, we are talking about plants. Okay. So this, as you can see, is tulsi. This is the haritaki fruit. So one of the first steps is correctly identifying the medicinal plant, or having the correct plant as is mentioned in Ayurvedic text, right? And in fact, of course, I'm no expert, but what I have read and heard is that many times there is something very specific written, that this particular type of plant which grown in particular conditions. So from modern biology, how can we match that description? Right? So first of all, we need to know we have the correct plant. Now, in olden days, people use more of phenotypic markers or characters to identify plants. But many plant biologists, in fact, tell me that this can be very confounding if the plant is not growing in the exact same condition. It may have some slightly altered phenotype. It may not look the same. For instance, something grown in a very saline environment, that same plant, if you put it somewhere else, it may look different. So it may, confuse a person. Is it really that? Right? So then came the next level of biomarkers. But today, we are at a stage where we can start talking about entire genomes, right? So we have this paradigm, and I'm sure you all know this basic biology, where you have DNA going to RNA and protein, right? So we are in the era where we are talking about entire genomes, right? So if imagine now at our disposal, because sequencing has become much cheaper than what it was, let's say 2 decades ago when the first human genome was sequenced, it's much more doable. So we can think of a time where we systematically identify the important plants, make a directory, and systematically do this genome sequencing. And once we have the genome, we will know what kinds of genes are there. Now, somebody would argue that why do we need to have a genome? We can directly look at the secondary metabolites. Usually, those are the ones that play a very important role in the medicinal properties. But what is important is if I have a handle of the DNA or the genome, I already know what the plant is capable of doing, right? So I know the potential of the plant. And it may so happen that when a sample is taken, I may not get the metabolites that I expected because of the growth conditions. Once I have a genome, like a gold standard for a particular plant, then I can think of what is known as transcriptome. So what are the RNA that are expressed in a given condition? So then I can get a handle on, oh, now I know if I grow this plant in so and so condition, I have this transcriptome that is there, which will ultimately lead to this metabolome, right? Because if we are very interested in the secondary metabolites and we have a detailed insight into all of this, then we have a much better control and standardization in making products. So I think one of the biggest areas that can have a lot of computation is plant genomics. Let me explain one more time. So if we have the genome, we can work out all the metabolic pathways, right, that the plant is capable of having in principle. It may not mean that everything will be expressed under all conditions, but by doing very systematic studies under different conditions, let's say we intend to grow this particular XYZ plant in such conditions, we can get a very beautiful scientific handle on this condition. What is the transcriptome? What pathways are likely to be there? And what is the metabolome that we can expect? So we have a much better handle on the exact details of the plants. Okay. And computation, of course, is an integral part of doing genomics, transcriptomics, or metabolomics. So I would say one of the very, very crucial areas that can revolutionize Ayurveda is by systematically doing this type of analysis. And I would like to point out that in the US, there is already a medicinal plant genomics resource, and there is an initiative where they have begun looking at very systematically, taken a few plants. They have started looking at genomes, transcriptomes, and metabolomes of plants. So I'm not saying that we don't have it already in our country. Some plants have already been sequenced, but they are very, very handful. There is no systematic effort to create an index of all the important plants that are related to Ayurveda and systematically do the study. Very recently, the Chinese have also announced 1,000 medicinal plant genome initiative. They also have a database which they have announced. Right now, it's not populated with too many sequences, but they intend to get there. So I think what will be very, very crucial and important for Ayurveda research also, where a lot of computation will be involved in putting together the genome, because I won't get into too many technical details because At some places, it may be de novo assemblies, for instance. So this requires a lot of computational pipeline. Then from that genome, if you want to work out the metabolome, again, there's a lot of computation involved. So this would be the right time to carry out such type of massive exercises of systematically creating a nomenclature and doing sequencing, and not just genome, but then also the transcriptome under conditions where we want to grow it, and of course the metabolomics. So there are some databases, this is a very, very recent review that talks about secondary metabolites that are there in plants. And like I said before, it's not just Ayurveda, but across the world, there are many different traditional medicinal systems, including even the Mexicans, the Africans, the Chinese, they have their own traditional systems. So you can see that there are some traditional Chinese databases. So I'm very happy that we have our own Indian initiative called the IMPACT, which was, I think it's not more than a couple of years ago. So this database is available, it's online. So what it does have is around 1.7 thousand small molecules, or these are secondary metabolites, and they have been deposited in the database. So I think you can search. I have had a cursory look at the database. So you can search by plant. For instance, if I say Terminalia chabula, you can go there and select that from the drop down, and it will give you a listing of all the different small molecule or secondary metabolites that are there in Terminalia chabula. And I think they have given nomenclature as per plant type also. So what is there in the fruit and so on. Many times what people tend to do is, if they don't find information like this online in a database, then they do their own experiments. But what I would like to say, that when you do experiments, you are limited by the kind of experiment you're doing, because if some of the metabolites are volatile, for instance, you'll never capture them. Now, the beauty of computation is, if you have the metabolome available to you, you will know what you can and what you cannot capture by your experiments, right? So you know what to expect, and if you know the properties. And again, I must say that when we talk about use of computers to evolve or to understand Ayurveda better, you need to know a lot of bioinformatics, some of chem informatics. So because I come from a department that is bioinformatics and my training is such, these terminologies come very naturally to me. Okay. So if at any point there is something I need to stop and explain better, please let me know. So like I said, this database, I'm very happy that we have this solely dedicated to medicinal plants related to Ayurveda in the Indian context, but this is not a be all end all. And there are many other plants, let's say, that are not there in impact, but are found in other databases, but are important to Ayurveda. Okay, so there is still a lot of scope for development of such resources that can be done to, have almost try to have more of everything. Okay, so our understanding of how things work depend on how best we capture what is there. So if I want to try and understand how this works, for instance, let's say even one fruit, until and unless I understand every component in there that is capable of contributing, my work will be incomplete, right? So the first type of let's say effort that needs to be done is, A, am I working with the exact right plant and the right fruit grown in the right condition as prescribed? B, am I able to capture everything that is inside of that plant part, whether it is the leaf or whether it is the root or it is the fruit, right? And for that, if I have a system that is exhaustive, for instance, like I discussed, then that will make our work more robust. Okay. So now I move towards what is very commonly done. And I must say that first effort was done by our neighboring department, by the group of Dr. Bhushan Patwardhan, where they tried to use a systems biology method to try and understand how these things work. And so this is called network pharmacology. I'll just spend a little time to tell you what basically it comprises of, right? So like we discussed that Ayurvedic or herbal medicine can be a formulation. So it can have multiple components. Let's say not one, not two, but maybe three or four. So the first thing is we need to understand what is inside. And again, I'm saying this is where we may be falling short right now because we don't really understand everything that is there. Perhaps because we are limited by the data that is available to us. Perhaps we are limited by the experiments that we perform and the fact that those experiments are not able to capture everything. Okay. So right now what we are doing is either we do our own experiments. So let's say I have a formulation and I'm talking about the general literature that I have been reading and going across. That people either take the formulation, do different types of solvent extractions, perform LCMSMS or such kind of analysis to find out the secondary metabolites that are there, right? So again, there is a chance that perhaps something was missed. Some people scour the literature, right? They see whether anybody has already done experiments and what type of secondary metabolites have been reported. And then they create a compilation, right? So I have a list of, let's say, 5 or 6. This is, of course, just an example. This is not a real example. I've just picked up some random small molecule formats, right? So let's say, just for example, that this was the formulation, and I found either by literature mining or by doing my own experiments, I found some metabolites. The next step is, what does this metabolite work on, right? I try to figure out the targets, the protein targets, because our understanding so far, okay, is that based on even Western medicine, most of the small molecules, right? So if it's a small molecule, something like this, it must be working on some protein target, right? That must be upon modulation of which some physiological response is observed. So this is our understanding right now. So this is how we are fitting it in our understanding, right? So there may be many more things that we don't understand, but this is what we are presenting based on our understanding right now, right? So what we do is we take these ingredients, we try to identify targets, and I'll spend a little more time here. And then we say, okay, so these are the, let's say the red ones are the small molecules, and these blue ones are the different kinds of protein targets. And this is how perhaps this works, right? So this is like a model, which is referred to as network pharmacology. Why network? Because this is so different from single molecule, single target, single disease, right? You have multiple compounds working on so many different targets, maybe on so many different diseases, like we saw in this example, right? Of Trifala. So it's more of a network. We need a network to explain this. We can't have just one connection, right? There are so many connections. So it's more like a systems approach. But again, we are limited by our understanding. This is how we are fitting it because this is how we best understand it. Now, this is how we best understand it, fine, but we need to, those of who use this need to understand a couple of things. Is that you need to go a little bit more into details of bioinformatics, right? Now, once I have, let's say, some kind of small molecule, I'm going to say, what is the target or what can it bind? What protein can it bind? So there are two approaches of identifying targets, and I wanted to put this across because I read so much literature and this has to be known. So there are two ways. One is you identify known targets. Okay, so that means by experiments, it is known that protein 1 is a target or it interacts with this small molecule. Alternately, there are tools that give predictions. That means given the small molecule, what protein might it bind hypothetically. Okay, now these predictions may be good, may be bad. We don't have an idea, right? So one should primarily look for targets which have been experimentally validated. Okay, so PubChem is an excellent resource. PubChem, Campbell, BioGrid, PDB, Drug Bank, Binding DB, where there is experimental data available for small molecules. Okay, so one should ideally try to go with this. So the model you're building is not hypothetical. Then it means that, oh, these three phytochemicals that are found, indeed, it is known that these proteins interact with these small molecules by this IC50 or whatever, or KD or KM, depending on what the protein is, right? So this information you can get from different databases that compile bioassays across literature or as submissions from authors. Alternately, if you just can't find anything, perhaps then you can do predictions, but then it's more of a hypothetical nature, that network, right? Similarly, once you identify a bunch of proteins and you want to say, is it really now involved in diabetes or digestive disorders? Again, there are two types. One is that there are validated targets for diseases, which you can find in a couple of different databases like the therapeutic target database or drug bank, or there are literature associations where through literature mining, people have said that this target is associated with this disease. Okay, then it becomes slightly more hypothetical network. So when one builds network pharmacology and one wants to propose a molecular mechanism, it's always best if one tries to look at data that is validated experimentally. Okay, unless there is just nothing, then it has to be put across that this is a prediction. Okay, it doesn't mean that once you have done this network, that's it, that's how it works. A, one doesn't stop there. B, right, we don't know unless you are convinced that experimentally this is how it works. Otherwise, then you have to do experiments to show that this is the way it works. Unless network pharmacology is rigorously tested in experiments, it will never be accepted, right? So this is just a preliminary model that gives you an idea of how it might be working, which needs to be rigorously tested in vitro and in vivo. Then it completes the entire story. Okay. A lot of people also then use molecular docking and MD simulation. I very briefly I'll touch upon this because I'm kind of exceeding my time here. So what does molecular docking do? You have a protein structure. Either you have the structure available from experiments or you built the structure. And let's say you have some compounds which we refer to as virtual library. There are many different tools that are able to predict whether how this particular compound, let's say, will bind to the protein. Now, a lot of people do docking and they feel convinced that because I got some score, it must mean that, that this is how it works and it's successful, but it does not mean that. And I would urge everybody, those of you who use this technique, to use some dummy small molecule, do the docking and see what happens. If you take any small molecule in a relatively large or in a protein that has a relatively large pocket, you will get some score. Okay, so how can you make your docking rigorous? The way you can make your docking rigorous is by running controls. So is there already known compounds that are known to bind this protein? Is there available co-crystals? Is your docking tool able to reproduce existing experimental knowledge? Then the unknown that you have docked perhaps makes sense. Still, it is perhaps, again, this is only a model. Unless it is tested rigorously by experiments, this will always stay a model, okay, and never be accepted. Even in Western medicine, the entire pipeline of discovery only has in silico or this kind of approach is the very first step. Then you have to go to in vitro, in vivo trials, phase 1, 2, 3, and so on. Only then it is accepted. So people who will stop here being convinced that this is how it works, we must go a step beyond and we must have a lot of experimentalists who are also excited at doing this and actually test and show that it works. Similarly, people do a lot of simulations and I'll just run this because I think this is a very neat method. So molecular dynamics is nothing but what it does is a technique where you use Newton's second law of motion to try and see how molecules are moving and hence functioning. Okay, so when we go to, let's say, structure, I want to get a structure of a protein from the PDB. That's just a static structure. most of the structures you see in databases that you download will be just static. It's like I took your photograph. That's not how you are, right? So everybody is moving, doing things, and that's how they are really. So in reality, all our biomolecules, be it small molecules, the secondary metabolites, the targets they interact with, they're all moving. And if you want to understand, and this animation, I'll run it again, is of a drug actually outside somewhere in the solution, how it has beautifully captured the entry into the receptor. Okay, and no other technique, experimental technique can capture this as can molecular dynamics. But of course, you need to know a lot of theory for this. You need to know how to build this, how to apply this, how to interpret, but very, very important technique to understand. Let's go ahead. So there are, of course, several unanswered questions, even in this small thing that I just discussed. For example, right now, based on an understanding, we are still doing one-to-one, right? I'm taking one small molecule, identifying its targets, but we are not modeling the mixture. When the formulation is given as a mixture, what is its chemistry, right? Is the ionization state perturbed by each other? What is the role of the solvent? Right now in our model that we are popularly using, it doesn't address this. What about the degree, the edges that we are creating in the network, right? What does it actually imply overall on systems level? What about the experimental validation? These are just a few points I have raised. We need so much more to go ahead to progress further. Okay. Quickly, I'll also touch upon few other topics. For instance, the herb-drug interactions or the toxicity. Now we know the entire, let's say, pharmacology or pharmacy tells us about pharmacokinetics and pharmacodynamics. So when we take a particular medicine, it needs to be absorbed, it needs to be distributed. Some part of it may be metabolized by the liver, some part of it may be excreted. So essentially that is known as the ADME. So before deciding a dosage for a medicine, pharmacokinetic studies are actually performed to see what fraction of what is taken orally will be bioavailable or in the blood for systemic circulation. Okay, so this is basically ADME. So if I give two concomitant small molecules, now whether they are two drugs or two phytoconstituents or one phytoconstituent, one drug, how can they interact with each other, right? So one of the ways they can interact with each other is perhaps, for instance, I'll take the example of interfering with each other's metabolism, right? So what is the role of the liver? The role of the liver is actually to look at these foreign chemicals and to perform some small molecule reactions like conjugation or hydrolysis, break them down and throw them out of the body because it's harmful, we don't want it, right? But after calculating what fraction will be metabolized and what still goes in the blood, we decide the dosage. So let's say for medicine X, based on this known, we have decided a particular dosage. Now, when I give something else, let's say some herbal medicine, okay, and maybe there are some cytochromes, and there is a huge family of cytochromes. So I'll directly jump to this example. So, for example, very, very well-known case of crosstalk between grapefruit and warfarin has been recorded. So, warfarin is essentially a blood thinner. It's come from Western medicine. And it's known that if you take it with grapefruit, then you will have toxicity from warfarin. Now, why this happens is because warfarin is known to be metabolized by these two particular cytochromes. And the ingredients of citrus actually go and block these cytochromes. So if these are blocked, it can't no longer metabolize warfarin. So actually the amount that goes in the blood increases and hence you have toxic side effects. Okay. So how can one actually do these predictions in advance, not just for interactions, but in general to predict toxicity also? So there are many online tools and with different levels of accuracy. We've just begun looking at them. So for instance, there are some combined tools like Swiss ADME Admit Lab, which will actually give you some predictions as to which cytochromes they might interact with, what will be the bioavailable amount and so on. So you can actually do these predictions. So a lot of computation here is involved. There is scope for a lot more because none of these tools work at 100% accuracy. So in a priori, if we can predict, and once you have some red flags from these predictions, go back and check these interactions or talks predictions in experiments, and then only go and ahead for the prescriptions. This would be very, very beneficial. And last but not the least, I think I'm over exceeding my time. If I don't talk about machine learning, any computation overview would be incomplete. So I must say that machine learning is revolutionizing biology research in general, right? So what does it need, and what is the entire flow of machine learning or this predictive modeling? So what it requires is a lot of data, and we have a lot of data. In Ayurveda also, once we start smartly compiling and creating these databases and data sets, we will have a lot of data, and perhaps it's my limitation that I may not be aware there already may be a lot of data out there. Okay. So what can we do with this data? So ideally what we do, if I want to build a predictive model, let's say I have, I'll just take a general example of, I have 10 phytochemicals that I know that work for let's say disease X, and I know that 2 don't, or 10 of these don't work for disease X, right? So I want to then make a model to train my data that given some unseen chemicals, I need a predictor to say whether it will work on disease X or not. So I need some training data. So we call it, you have to have a positive data set. That means you know these compounds work for this. This is just a hypothetical example. It can be anything. It may, your question may be anything. It may be what parameters are predictive for Alzheimer's. It can be anything, right? So I'm just giving a very, very general example. So you need a positive data set and a negative data set. You need some class label, which means this will work for disease X, this will not work, to train your machine or you to train your network. Okay. So what is done is, if I have small molecules, I'm going to extract the features. So how does machine learning work? So the computer actually trains itself on features that are, let's say, present here, absent here. So then it can correlate. Once you have this, make this prediction. And that's how our brains also work, right? So this is actually simulating real brains. We train ourselves. How do I recognize girl versus boy, right? If I see girl, what do I see in girls that I say, oh, this is a girl? There are some features, right? Let's say long hair or wearing bindi. So these are features. So even machine learning algorithms need to be trained on features. So there are algorithms that are trained to extract features. A matrix is created. There are different machine learning algorithms. I won't go into too many details, like SVM, random forest neural network, so on. And you create a model, right? You can evaluate it. So on your new data, how well is it performing? If it is not performing well, you can go back, modify data, and so on. So essentially, the idea is that once you have a lot of data, then you can create predictive models. And this is done a lot in bioinformatics, cheminformatics, right? A lot of skin talks or so many of these are based on pure machine learning models. Okay, so with this, I think I'll stop here. I think I have given a brief introduction of many, many different things, like an overview. And I think need of hour is we need a lot of training modules on informatics that can be used. Like I said, if we want to know how we can do this better, we need to know the techniques better, right? And to do this, we need a lot of brainstorming and coming together of Ayurveda professionals and scientists. And I'm so glad that the organizers are doing this and putting both Ayurveda and scientists together. We need to have more hackathons. For instance, a hackathon can be announced for just modeling of these mixtures, right? The chemistry of mixtures. Many more conferences should happen to create awareness and more crosstalk. So then this field will progress. So with that, I'll stop here, and thank you very much once again.
Thank you very much, Dr. Joshi, for your insightful talk. I request participants, due to limitation of time, please write their questions in the chat box, and I request experts to please reply there. Okay, now switching on to the next speaker, we have with us Dr. Ved Priyaji Arya. She is a postgraduate gold medalist in biosciences, and she is doctoral in genetics from Rohtak University. Dr. Vedpriya Arya has worked extensively in the integration of statistical and technological methods of plant-based medicines. She is currently leading the Patanjali Herbal Research Department at Patanjali Research Institute, Haridwar. So over to Madam, Dr. Vedpriya Ji Arya.
So thank you so much, Prashant ji. It is nice to interact with you and all the audience. And I especially thank to Dr. Manali Joshi ji, who has given very insightful talk regarding the computational approach in the biological domain. And yes, we have our audience has several questions, and definitely they will put in chat box. And I request all the participants from Patanjali Research Institute as well to write your questions in the chat box as directed by Mr. Prashant Gupta. So before starting, I would like to just introduce very briefly Patanjali Research Foundation Trust. However, Patanjali is a very big name and each and everybody is very much familiar with that. So just I am showing some of the research highlights of Patanjali Research Foundation Trust. As you are much aware, Patanjali Research Foundation Trust, it is inaugurated in 2017 by our Prime Minister, Shri Narendra Modi ji. And we are having the ultra modern laboratories. And what is the main agenda or the basic funda? To convert the unstructured Ayurvedic and herbal information, which is in our ancient text, or you can say ancient traditional historical text, into the well-characterized Ayurvedic herbal medicine, which are scientifically validated, which are evidence-based, according to the modern practices. This is the major aim or the vision of Patanjali Research Institute. You can see this is the research strength and accreditations at Patanjali. We are having biosafety level 2 research laboratories, NABL accredited and QA compliance laboratories, DSIR zero certifications. We are following the CPCA guidelines, as well as we are having 250 beds Ayurvedic hospital, which is NABH accredited, and 15,000 medical doctors on roster, and daily footfall of near about 15,000 patients across India. Well, this is the research strength and accreditations. And coming to the Patanjali Research Foundation, so we are working in several segments like the herbal research, drug discovery and development division, painting herbarium, ancient documentation division, yoga research, and several sister or allied institutes. Why I'm saying this? Because we are working in diverse areas. Like we are from the ancient documentation because we respect our ancient literature, Ayurvedic literature, and through herbal research domain, we have given the World Herbal Encyclopedia to the society, as well as drug discovery development division. You can say this is the drug discovery development division at Patanjali, and which is again divided into 5 segments: in vivo biology, in vitro biology, herbal chemistry, microbiology, and clinical research. And you know, we are having the state-of-art infrastructure. Well, at present, we are following the disease-driven approach, and for inflammation, 32 medicines which are evidence-based with clinical trials have been launched. For metabolic disorders, cancer, neurosciences, and infectious diseases, and so on. We are the premier institute who have launched near about 40 medicines which are of GRIT series, and those results are comparable to the modern medicines, and these are evidence-based. This is the small snapshot of the cardiovascular disease. We have worked very hard for cancer, and some of the medicines which are shown over here. You are very much familiar, and very common medicine is the Coronil, which is a boon during the corona pandemic time to all of the individuals residing in our country. So what are the basic sectors? We are working on the herbal ward, conservation of phytodiversity, ethnomedicinal data preservation, and very, very important, World Herbal Encyclopedia, and we are working in several other domains of the training and the capacity building activities. Well, our phytodiversity division, it is accredited by New York Botanical Garden Index Herbarium. We are collecting live plants from the field. As you are seeing, Pujacharya Balakrishna ji is leading the team of scientists, and under his supervision, all the scientists are trained enough to identify the plants, to identify it taxonomically and preserve in the form of the herbarium. We are having a far approach regarding the ethnomedicinal database preservation. Well, we have done Center of Excellence project under the Ministry of Tribal Affairs, and at present we are running with the Center of Excellence Ministry of Ayush. This is the encyclopedia. It is very, very important work for all of us. Why it is important? Because it is covering the information of 50,000 plant species, which is compiled in 108 volumes. And very important point to note, there are 12 lakh vernacular names of the plants which are in more than 2,000 languages of the world. A very crucial work, very important work, which is again considered as the innovation in Ayurveda research, is the Vedic taxonomy. It is one of its kind, dear students, as well as all the scientists. Why it's one of its kind? Because in Vedic taxonomy, each plant has given a scientific Sanskrit name, and this name is based on the guna, dharma, and dosh, or you can say morphological characters, anatomical characters, and the properties of the medicinal plants. It is one of its kind work, and at present, under the divine guidance of Pujacharya Balakrishna Ji, a Vedic taxonomy of 350,000 plants has already been done, and it is regarded as innovation in Ayurveda research by National Commission of Indian System of Medicine. Well, we are documenting first of the time medicinal history of the world. We are covering 962 healing practices. We are covering 1,300 diseases, and we have used near about 8,500 bibliographic resources in the compilation of the medicinal history of the world. Well, there is the Ancient Manuscript Preservation and Conservation Division, where we are preserving and conserving the ancient manuscript which are in Tamrapatra, Bhojpatra, as well as in Tadpatra. And this is the recent scientific information that we have conserved in our institute. Well, coming to the present topic, International Conference on Achieving Holistic Health through Ayurveda along with Advanced Technology. I'm very excited to share a very short glimpse of this particular conference with you. Well, it is planned from 1st of May to the 3rd of May in this year. And I am glad to say that it is under the Center of Excellence project, Ministry of Ayush. All dear participants, I also welcome you. I invite you to join this conference by your physical presence in Patanjali University in Haridwar. Well, what are the conference themes? We have worked very hard, and I especially thanks Dr. Ashwini Kumar from Patanjali, as well as Dr. Girish Ji, who guided us several times. And because before this particular webinar, we are having several sessions with them, several brainstorming sessions, and we have worked very hard to improve the domains and the theme areas. The first conference theme belongs to the Ayurveda for physical health and wellness. It includes because Ayurveda is not a simple concept. It includes the diet. It includes the ahar, vihar, dincharya, ritucharya. It is managing the lifestyle disorders through the asanas and pranayams. So we have special sessions for this particular theme. Scientific aspects behind the Ayurvedic rejuvenation therapies for improved immunity and metabolism. We are having the phytochemistry aspects of Ayurvedic nutraceuticals for support, sports and the fitness enthusiasts. So dear all participants, I would like to clear over here. The first theme, it is dedicated to the integrated pathy, or you can say holistic approach of the traditional medicinal system. We believe that naturopathy, Ayurveda, yoga, homeopathy, Unani, as well as the acupressure, acupuncture, and very, very important, the diet therapy. These are the boom for the mitigation of any sort of diseases. The second theme, it covers Ayurveda, a ray of hope for mental health care. My dear participants, as you are well aware, for professionals, and not only for professionals, for daily workers, we are having the mental stress all the time. So we think this is the second theme, combating stress through the integrated pathy, inculcating resilience through yajopathy and yogic practices. During our research, we have found that yajna, which is also known as Agnihotra, and sometimes in foreign, it is also known as the Homa therapy. It is having the equal benefit for treating the mental disorders, or it is very important for the mental health care, for the cognitive behavior, for memory enhancement through the Ayurvedic Rasayana therapy, emotional intelligence through the Ayurvedic lifestyle. So this is our second thematic area. The third thematic area, it is very, very important and novel for all of us. It is the artificial intelligence and data analytics in research. So it includes the surgeries, it includes the AI ML, it includes the data analytics, and it includes the technological driven approach for the phytoconstituents in medicinal plant. I would like to share some of the glimpse of the Patanjali Research Foundation Trust. My dear participants, we are working in bioinformatics since long. We have the very good kind of research in the form of the triple negative breast cancer. We have published more than 10 papers and a complete book with the Bentham science in the triple negative breast cancer. And not only in this particular domain, we are working in several domains in the bioinformatics and the docking side. Second, very important is the hospital management system that will be showcased during this particular event in the conference also. Hospital management system, it is very newest one to all over India. And I'm very proud to say that in Uttarakhand, our hospital management system, which is developed by the Bharwa IT Solution Company, which is a sister or allied company of Patanjali Group, has already been implemented in Ayurveda hospitals of Uttarakhand. This is an achievement for us, and we are now going to establish it for all over India. Well, this is the organizing committee, our gurus, chief patrons, Revered Swami Ramdev ji, Pujya Acharya Balakrishna ji, who guiding us all along the way in this particular journey. We have our patrons, Professor Mahavir Agrawal, Pro Vice Chancellor of University of Patanjali, Dr. Sadhvi Devpriya ji, Dr. Anurag Varshney ji, Dr. Anupam Srivastav ji, Dr. Anil Kumar ji, and Dr. C.B. Dhanraj ji. I'm very, very proud to say we are having the organizing secretaries, a great team of coordinators and the members, and we are having the honorary advisory members. I again thank Dr. Ashwini Kumar for working as convener, and not only convener, he is executing all the tasks with the help of organizing secretaries and senior team leads of Patanjali Research Foundation Trust. Well, these are the important dates, abstract submission, and because these are open from since January, and the last date is 10th of April, so all the participants should hurry, as well as we are having the confirmation, and further we are having the guidelines for abstract submission, and these are the some of the details regarding the registration, as well as we have also guided in brochure that how you have to reach University of Patanjali, and some contact details are also given over here. So with this, I would like to thank all of you, and I invite personally all of you, and please take it as my personal invitation as well, and thank you so much.
Thank you, Dr. Vedpriya Arya, madam. So I think we should inculcate this information to the larger extent. So I request participants to please share the information to their social groups as well as to the Facebook, so that a wide information should be sent to each and every stakeholder of Ayurveda and the research. Now, coming on to the third part of this webinar, we have a very energetic, honest, and dedicated researcher of the field, Dr. Saket Ramji. He has an extensive research in field of Ayush informatics. Dr. Saket Ramji is very well known for his work on the Namaste portal and digitalization of Samhitas. He has worked with National Institute of Indian Medical Heritage for more than a decade. He currently works as an officer on special duty in the Ayush Grid, Ministry of Ayush, Government of India. So I request Dr. Saket Ramji for the talk over technological initiatives in Ayush sector. Over to Dr. Saket Ramji. So,
yeah, Dr. Prashant and all the dignitaries, Very good evening to all of you. Actually, I need just a couple of minutes. I have came out and I'm actually now talking to the mobile. Just give me 2 minutes. I will switch to the laptop and I will present. Is it okay?
Sure, madam. Sure, sir.
Thank you, sir.
So meanwhile, we can have 2 to 3 minutes for direct question to our speakers. If any of the participant had any query, they can ask
Prashant sir, I have a
to
question.
Dr. Joshi or Dr. Vipriya, madam.
Prashant sir, I have a question. First of all, thank you,
Uh,
madam.
Meanwhile,
Prashant sir, can I ask a question from Manali?
with us, who is going to give a concluding remark over this webinar, Dr. R.S. Rajanji.
Yes, Dr. Gupta.
I rejoined, sir. So am I
Hello.
audible?
Yeah, you are audible. Am I audible to you?
Yes, sir. Yes, sir.
Yeah, okay. So I'm listening from the beginning. So there's no problem.
Okay, that's great, sir. That's great, sir. Actually, I'm just giving a brief about you. Dr. Rajan is a retired brigadier from Army Medical Corps. After heading the Department of CTVS, Cardiothoracic Vascular Surgeries at AFMC Pune, he trained cardiothoracic surgeons of Army, Navy, and Air Force for over 14 years. And he is known for his passion for teaching, both undergraduate and postgraduate, and paramedics. He is engaged in community health services for last 20 years. And we are very fortunate that he is with us in this webinar and in the series ahead also. And we are also working on the special issue. So that's all from, I think, Dr. Saket Ram, sir. Are you ready with your presentation over laptop?
Yeah. Just a second.
Dr. Gupta.
Sir, can I ask a question from Manali, madam, if you permit?
So, just a minute, madam. Dr. Brigadier R.S. Rajan, sir, is speaking something. Please, sir.
Dr. Gupta, I just wanted to know that it will be all right if I give the comments after all the three speakers have finished, isn't it?
Yes, sir. Yes, sir. We expect after 3, sir.
Yeah, okay, fine.
Please question participant, please.
Manali, madam. First of all, thank you so much, madam, for enriching us with your gigantic knowledge. I have a question regarding the transitions of the DNA and RNA molecule during the MD simulations. See, nowadays it's very much in trend that people carry out simulations or the docking study with the proteomics. Okay, we do study with the protein level. But when we move from the protein towards the DNA or RNA level, that's the genomic markers. So can you please discuss about the transitions that happen in the DNA or RNA molecule when we carry out docking study or the simulations?
What exactly do you mean by transition?
See, what I am supposed to say, what I want to say is actually when any of the ligand is binding to protein, DNA or RNA molecule, the structure will not remain the same. Some sort of the transitions will be happening. If it is happening at the protein level, it's fine. There is no problem with that because if at the translation level, it won't create any genetic difference. But if there is any sort of transitions regarding the DNA or RNA level at the replication or the transcription level, will it create a problem at the genetic level in the other genes? So I just have a question regarding that.
So there are known drugs that are DNA intercalators, for example.
Recording
I'm
in
not
progress.
talking about the
Maybe we can have this later. Dr. Saket Ram.
sorry, madam, your voice got interrupted.
I think Dr. Saket Ram is ready, so we can take this up later.
Okay, madam. Thank you so much.
We'll share the email also at the end of the session, and participants can directly mail the queries also. So, Dr. Saket Ram, sir, kindly unmute yourself. Okay. So I request that organizer please unmute Dr. Saket Ramji. I request host to kindly unmute Dr. Saket Ramji.
Oh, yes, yes. Yeah.
Sir, you are audible. Kindly continue.
Yes. Please also allow me to share my screen. I think screen sharing is disabled. Yeah. Can somebody allow me to share the screen? Uh, yeah, before,
I request.
yeah. Can I share
Now
the
you can
screen?
share. Now you can
Yeah.
share.
Thank you, sir. Hope the screen is visible.
Yes, sir, you are visible.
Yes. Yeah. Thank you all. Thank you the organizers for letting me just share some of the developments and initiatives by the ministry and also overall as a sector. So in contrast to my earlier two speakers, I'll be presenting a very generic thing. It will be talking about laying out foundations for the technology in general and digital technologies in particular. Prashant, am I audible?
Yes, sir, you are audible well.
Yeah, thank you so much. Thank you so much. Yes.
I request participants to kindly mute.
Yeah, you all know technology, it's application of scientific knowledge to the practical aim of human life, or as it is sometimes phrased, to the change and manipulation of the human environment. So we have come a long way. I mean, I'm not to say from simple Yayavara to Gramya life, there's a big transition, and along with we have come up discovering a lot of tools to make our life better. So that's a general paradigm. And here, as I said, I'll be focusing more on the digital health platforms. When I say technology, actually, the presentation was supposed to include the technologies. For example, we use Yantra and a lot of Shastra and all. Of course, we have a very great legacy. Ayurveda was never averse to adopt technology. I just want to put that point in perspective, and also the very foundation of Vata says Vayus Tantra Yantra Dharaha. So Tantra Yantra are like a sort of inevitable thought process in Ayurvedic framework itself. So in that sense, what are all the technology is developed or say we are going to develop is very much within the ambit of Ayurvedic domain of knowledge. So we should not think that this is something alien. Of course, as long as it is not a Ati Yoga, say, or a Mithya Yoga. So here I will be talking mostly about the digital health platform, which from the sector, what we are doing, from also from the ministry, what are the kind of efforts we are doing to create this platform basically. So it's a big task. It's a multiple component thing. You can see on the screen. It has different kinds of requirements, and many people are required to contribute to it so that this health platform is a meaningful entity. Before we go further, I have few definitions. These are the standard definitions by WHO. So I'm not going to read them. I'll just pause few seconds so that you can read by yourself, and I'll go further. So now this digital health is becoming a normative term now. So it covers the concepts like e-health and all of them together. Now we started using this term called digital health. Another important term is digital health technologies. So this is very important, and it's a recognized global phenomena. So under SDGs, 2030 SDGs. So this is very important component, and so we have to align with it so that we don't lag behind. And this transformation happens at multiple levels. I'm not going to all the details. My earlier speakers have touched few aspects of it. So from the analog to digitization, so we have come a long way. So sometime back, we used to have the phones which we used to dial, and now we have all the cell phones. I mean, that kind of transition we are all gone through. And in the medical practice also, it is happening. And Ayurveda, I would say Ayush sector is also very much part of it. So very interesting term I have found when I was going through this glossary. So now this, what happens with the application of digital tools. So now you see, for example, if you have to do a proper trial, you have to have a proper site and you have to control a lot of things. But with the digital acumen, you can even have a digital trial which is siteless. In fact, we have seen during COVID this happening. Some people were like getting treated in quarantines and other places, but they were further monitored properly, and we could also publish very good papers. So it is not in the futuristic sense. Already we have done it, and this is the kind of transformation that digital technology allows us. I mean, it in fact erases the boundaries and increases the scope like enormously. And coming to segment, of course, though I have put this as Ministry of Ayush, please read it as the Ayush sector itself. So we are not in isolation. We work with many other other counterparts, and of course, this is only an indicative list. So this is hugely networked system. We are a part of a big entity, and our role is very important when we think about a platform. So we should consider many factors of which are depicted here. So to envisage this, to have this digital health infrastructure for Ayush sector, basically. So our project has been conceived, which is called Ayush Grid, which is a sort of integrating platform, not just within the ministry or say state governments, all the private and other stakeholders also. So I'm not going to read all of this. In summary, I have told you about the very function of Ayush Grid. So basically, it's a networking platform to bring all the stakeholders together and provide the digital infrastructure required, maybe in terms of policies, maybe in terms of bringing the good technology, or say helping people to realize their own potential and bring forth the best technologies for the sector, overall sector. Anyway, these are few statistics about the efforts done by Ayush Grid so far. So I will pause a few seconds in this particular slide. So we must remember that earlier it was mostly a hospital-centered, also a healthcare-centered health system, but now we are moving to the citizen-centered, vertically, horizontally. Now the citizen is more important, and the whole flow of information is through this. So recently, you must be aware that Health Authority of India has come up with Abha ID, which is going to be the pivotal like transaction point for all the health transactions in the future, like Aadhaar for financial transactions and direct benefit transfer. It is going to be the Abha for everybody. So personal health records are going to be the pivotal and not the hospitals being the custodian of the records. So that paradigm shift you must remember, and to make it happen, so we should actively, proactively participate in this digital ecosystem. So as you know, for this repetition of the same slide, the patient record becomes more and more important, and we are giving the power of record keeping to the person, individual now. So that's a great development, and actually it's sort of a big leap of faith which India has taken in these crucial times. And I mean, as one case study, I want to just project few slides about Namaste portal, as it is one of the early initiatives in enabling this kind of a digital transformation. So where, as you've seen in the first slide, if you see the terminologies are the most important part to create a digital ecosystem, and this was one of the early attempts. Before that, many attempts were made, but this was the one which was which could become public face and steered some other developments quickly in last 5-6 years. So I'll pause on this. Namaste portal touches many aspects. It is a work in progress, but as I said, it is basically an ecosystem development platform. So already it has enabled the development of standardized terminologies for Ayurveda, Siddha, Unani. We are now about to complete work on the ICD-11 codes, and also it has steered the BIS to come up with some of the glossaries, and many other things are happening. Now it is being used as a part of one health management information system within the ministry, and now we are going to scale it up to other agencies also. Of course, I'm not going to delve into these slides. Anybody can go to this Namaste portal and see this yourself. So I'm just skipping these slides. So this is the terminology part, and there are many use cases. Already this is being used in different situations, like sort of enabling more and more clear data collection aspects. So this is about Namaste portal. This is international terminology is recently released. You can go to WHO website and download. Basically, this is going to be the entrance point. Don't think that this is a comprehensive terminologies. It is a simple 3,000 to 4,000 list of terms in simple English, so that a foreign reader, a foreign audience also can have a basic introduction to Ayurvedic system and start, like sort of getting into the system If they want anything in depth, they can always come back and learn more about it. But basically, this is one international attempt where we could actually come up with a document for a non-Sanskrit or say non-Indian reader, so that they can relate to Ayurveda and get enthused about this. Of course, CKDL is one of the very oldest platforms where they try to digitize. Of course, right now the scope of TKDL is little restrictive. It is only available for these patent offices. But I heard if everything goes fine, I think with certain limitations, useful for researchers and others also. Dr. Shujani Madam was appraising. Hopefully, very soon we'll get access to this. It is a big repository of drugs and with the prior auth information. And this is one more initiative, IHMIS. We are developing this as a model health information system so that others can emulate, which has a double coding provision, which has proper system for prescription, and the ABA integration also completed. This is one of the very initial health recording platforms, electronic record platforms, which is completely Aadhaar enabled. And we are going to showcase this in forthcoming Health and Wellness Group G20 meeting in Goa. Then we will make it public. Of course, there are a few other things. Information Hub, Ayusoft. Ayusoft actually is a product of Pune. This is the first platform to digitize Ayurvedic assets, and we all got inspired with this. And in fact, personally, I'm very much a fan of this particular software, actually, which has prompted us to develop Ayurveda ebooks early in 2006 and 7. And a part of it, we could now digitize and make it public in a website format from CDAC. We could get permission and we could change. And the Prakriti software is also in progress. I think shortly we'll be able to give access in the public domain for this also. Of course, many people must have seen the Ayush Research Portal. One of the very good collections, around 30 to 40,000 article collection is there. We have this clinical case repository. It has not kickstarted much, but as an extension of it, Amrita Institute has developed some cell for the discussion of the clinical case records, especially clinical case reports, individual cases. I think that is already launched, and you can also check with Ayur Cell. That's a good initiative by Amrita in collaboration with Rashtriya Ayurveda Vidyapit. We have Namaste Yoga application, Vibreak application, Ayush Sanjeevani mobile, GIS portal. Admission portal is there. Ayush Next for students. There's an NGO portal. These are all for the ministry's internal use, of course. National Ayush Mission portal is there to track public health initiatives. And of course, these are the early attempts, as I said, the ebooks. And now, not only that, we have a very good translation online, Charaka Samhita online. It's a very good content developed collaboratively, thanks to Basishter and Yogesh Duel and others. It's one of the very interesting works which I could come across. I recommend people to use this. This is completely online and very nicely, well curated content. So these are the important steps to have this digital ecosystem. I mean, not just having any random application. These are all well studied, well thought of, and done by the groups of people. We have M-Yoga app with WHO, which has released, and this is being used very nicely in India and elsewhere internationally. And there are also other applications, nice applications. Just these are some examples. Don't think I'm promoting any one of here. Some of the very interesting things, whichever I could curate, I'm just presenting here. Dravya app is very interesting, a nice group. They have compiled, and at a click of the button, we have very good information available for individual drugs, substances. And there are also some AI initiatives now going on. Recently, this Ayur AI, they could get some award, AI Ayur kiosk, something called Ayur Rhythm. But these are all basically person-centered. Of course, word of caution: we need to be vigilant and examine these products. What kind of logic they are using, what kind of black box design they are using. We must be familiar with it. Otherwise, again, there may be a confusion in terms of the content validity and the kind of interpretation they are doing. So, so quickly, I'll wrap up my presentation by a few comments or say observations from my side. So now, digitization is inevitable. I mean, we can't do away with it anyway. But when we do it, let us understand and do it. I mean, that's the one buzzword which I always keep reminding myself, and also request my audience to remember that. So now, it's almost 75 years of our independence, and also 75 years of WHO's contribution to public health. And we are a part of it. Ayurveda, especially in Ayush systems, we have proved ourselves worthy in terms of management and also helping people get rid of COVID. After that, now the barrier of non-communicable, communicable, whether we'll be able to deliver or not, no such confusion exists anymore. We can deliver, and we are going to deliver, and we are also going to update ourselves this digital health platform and be able to abreast with the mainstream, so-called mainstream system. So one last initiative I would like to appraise to our audience that now with the Association of International Telecommunication Union and WHO, so we are part of a big discussion that's called benchmarking of AI in traditional medicine. Of course, Ayush is a part of that. So now I'm happy to inform that our team from India is leading it. I mean, no other country. Now we are driving it. I'm the topic driver for this, and thanks to Rajesh Vaidya Kotecha sir, he has allowed us to be part of this big discussion. And I'm happy to inform you that just last week I was in Cambridge, MIT and Harvard. There was a 4-day conference on that, so we have attended, and now we are going to have this policy draft very soon. So two things I want to mention, that the efforts are going on, and the inputs for this initiative are welcome. So this digital ecosystem will not be done by ministry in isolation. That is impractical to think of. So we request that you be part of these discussions and give us the input so that we have a robust infrastructure available for us, and we need not wait for anybody's validation in that. Externally, we are enough equipped. For example, if you take Patanjali or other organizations, they have done so much within their organizations. Like, for example, Savitribai Phule University, Girish sir team and Patwardhan sir team. You have created, in fact, much ahead of ministries' initiatives. You people have done a lot of work. So now we are here to embrace that and bring it onto a single platform and make it useful for the entire country, and ensure that we use this for the effective translation of whatever the wisdom we have to the public good. So with this, so I would like to sum up with these few slides from WHO, Digital Health Initiative. So we should now start thinking about proper investments into this. It is very important, and we should make youngsters as a part of it, along with the seniors. Second is, so finally, I would like to say there are no digital quick fixes for young people's health or anybody's health for that matter. So they work best when they are part of the existing health system. So we are not creating a pseudo or a super system here. We are going to strengthen the existing system and make it better. That is the final tagline. So to design sustainable digital solutions, young people's health, listen to them. Why I'm starting with young people here is that, so generally, I think we have two problems. One is ageism, and one is ignoring the young people's wisdom. So I think we have to bridge the gap between, and somewhere take a madhyamarga, so that we get the best of these two universes. So we being a legacy system, so we have more responsibility to bring on. In fact, when I say this, we have to take into confidence of all of our colleagues, irrespective of age groups, to make our products better. I mean, because we are going to directly talk to the individuals. Those individuals, again, each individual is very different. So to cater to that, we need to have a proper brainstorming, proper creation of persona, then design the application. If I think it is going to be right for certain utilization, it may not be the case at all. So I should consider others' views, be patient to do the homework, then come up with a proper product. Otherwise, most of the products we develop, but they're of not much use. I've seen many good, like for example, Triskanda Kosha. I'm taking name purposefully. It's a great work, but it's a gigantic work that many people are afraid of using it. So we have to think of something from the user's perspective also. That happens only with the proper dialogue. In digital era, that's all the more important. So with these few thoughts and information, so I would like to say thank you, and I'll open the session for further discussion. Thank you all. Namaste.
Thank you very much, Dr. Saket Ram sir. So participants, we request to kindly write down their questions in chat box. Maybe due to short of time, we are attempting this step. Now I request Brigadier Dr. R.S. Rajan sir for concluding remark.
I hope you can see me and hear me.
Yes, sir, very well.
Okay, all right. So thank you very much for inviting me to be part of this exciting webinar, and thank you for moderating it, Dr. Prashant Gupta. Overall impression is that it is very, very encouraging to see that so much is happening in the field of Ayurveda. It's very encouraging, promising, and exciting. Having said that, let me come to the individual speakers. As an army man, I would like to keep my comments brief. Dr. Dr. Manali Joshi spoke on an excellent topic. She really outlined where we are, opened our eyes to what are the possibilities with modern genomics and pharmacokinetics, network pharmacology, molecular docking, and so on. And she also concluded with some unanswered questions. And so it was a very exciting talk, and I found that she was talking the language which is acceptable to many people, because she pointed out very clearly that to be accepted in today's world, whatever we claim should be backed by modern scientific methods, whether we like it or not. I think that's the great message that Dr. Manali has given, and I would like to congratulate you for that. But I have some worries. The first worry is that, yes, we are working in on very important topics, combining modern methods, including artificial intelligence and digital technology, with the ancient problems of taking the various plant products and all that. Is this being disseminated along the length and breadth of Ayurveda world itself? Or is it some kind of an isolated work going on in some good research center? Because my humble reading of the history of medicine, history of modern medicine, if you take, people who set forth new ideas were ridiculed, were condemned, and pushed to a corner by the traditional practitioners of so-called modern medicine at that time, like in the case of anesthesia, antisepsis, and so on. Thank you. And therefore, I find that it is very important in research to take people along with you. And the people include people within Ayurveda and people across on the other side, meaning the allopaths. And if you want to work in silos, then you will end up with some very, very useful conclusions which are not used by others. So that is the first comment I have. The second comment I have is, I'm quite worried. Dr. Manali is doing excellent work, and she was quoting already the Chinese are doing a lot of work. The US is doing a lot of work on the basis of our experience of the past, especially with reference to basmati rice and turmeric. We should be very careful about our own research not being hijacked by these people, because this is an era of digital technology, and it's very, very easy to take away what you're working on and claim it as their own. And then if they just claim it as their own, I have no problem, but unfortunately, they will commercialize it also. And the third worry I have about Dr. Manali Joshi's excellent work is, I hope enough people are interested in this kind of work, and I hope there is enough funding forthcoming for this kind of work, because it is yeoman work, and I wish her all the luck. I am in Pune, so I'll meet her and sort of take up these issues subsequently. The second speaker was Dr. Vedpriya Arya. I found that she gave a very lucid account of not only the Patanjali Research Institute, who are working diverse areas, created the herbal encyclopedia, the Vedic taxonomy, and so on. And she also touched upon in detail upon the International Conference on Ayurveda and Modern Technology from 1st to 3rd of May, and the various themes which are going to be discussed. It was very educative, and I'm sure a lot of us are looking forward to it with great interest. I have only one request, that have they thought of involving leading allopathic physicians in this conference, or are we going to work in an isolated fashion and say that we don't care what they think? Because by my own little experience, I can say a time has come for us to realize that allopathy needs Ayurveda and Ayurveda needs allopathy.
Definitely.
They have to work in a symbiotic relationship and not in an antagonistic relationship. And if both sides realize this, it'll be a great thing. Particularly, I find while allopathy finds ready acceptance by the educated urban middle class, the educated urban middle class, which are the opinion makers of society, have to accept Ayurveda also as part of the healthcare system. And that cannot be done by fighting each other, but by working together. And I don't think there is any conflict of interest in this, because ultimately what we want is the good of the patient. Whatever is good for the patient, we should be ready to accept it. And that's what is the secret of the progress of modern medicine. They were ready to accept what is good for the patient, and what they were doing till now was bad for the patient. They threw it out, they jettisoned it without any regret. I find that kind of attitude should also come into Ayurveda. The talk by Dr. Saket Ram Tribula, as an old man, I must take the liberty of telling he tried to tell us too much in too short a time. And I think they missed a lot of good things he was trying to say. I know there is a lot happening, but then, as they say, it is the work of the genius to tell us in brief what is happening so that we can follow. But whatever I could follow, one or two comments I would make. He talked about Tantra and Yantra being known to ancient Ayurveda and acceptable. I think that kind of approach does not wash with me, because I find that Tantra and Yantra may have been in vogue in Charaka's and Shushruta's time. But what happened in between for many centuries? People didn't question. People didn't debate. People accepted. Even now, recently, I know there are many senior Ayurvedic physicians who don't want to question anything which is written in the old books. That is not being acceptable to Yantra and Tantra. I think so. There must be an attitudinal change. I'm not talking about Dr. Saket Ram's attitude. I'm talking about the attitude of Ayurvedic physicians as well as allopathic physicians, that they should have the readiness to accept the scientific method. And you all will agree with me that Ayurveda, which was at its peak in probably the few centuries before the present era and the first few centuries of the Christian era, it has declined over time for various reasons. And those reasons have to be gone into. We must learn from those mistakes and make sure we don't commit those mistakes. For example, too much of reverence for old texts. What is called for is irreverence for the old texts. What is called for is questioning them and trying to put them in the crucible of science and trying to prove them. If we are able to do that, I think then Ayurveda will find ready acceptance. He also, Dr. Saket Ram, talked about standardization of terminology. Highly called for. High time we did it, and I'm glad they're doing it. But standardization of technology should not stop with, standardization of terminology should not stop there. It should extend across, not only standardization of terminology, I would say standardization of syllabus of the Ayurvedic medical colleges across the country, standardization of the examination system, standardization of the training system, and so on. So standardization is the need of the hour, but its scope should be enlarged and applied to many other areas in Ayurveda if we want to speak the same language in the country. That means what an Ayurvedic physician talks in Haridwar should be the same as what the Ayurvedic physician in Kottayam talks. If they're talking two different languages, they can never meet, though they are following the same system. So this does not happen in modern medicine. That's one of the strengths of modern medicine, that if I say hypertrophic pulmonary osteoarthropathy, it means the same thing here and in Boston and in Tokyo. So therefore, we should try for more standardization across the board. And I believe there is a lot of dissemination, trying to have the dissemination of information across to foreign audience. Yes, that's a very welcome thing. But have we disseminated enough within the country? That also we have to think about. And unless we disseminate within the country, it will become something which is the curiosity of a few interested foreigners who will be out to exploit this for their own benefit, and our own people will be left out of the beneficiary, out of benefiting from this system. He said, I liked that comment, which was very revealing. He said, during the COVID pandemic, we proved ourselves. Do we need to prove ourselves? What does that mean? That means you feel there is a need for Ayurveda to prove itself. I think we should work from a conviction that Ayurveda has many good things to offer, and it should be part and parcel of our healthcare system. I appreciate the last comment which Dr. Saket Ram made in his presentation. He said, whatever we do must fit into the existing healthcare system. I think that's very important. So you cannot wish away allopathy any more than allopathy can wish away Ayurveda. So what is good for the patient, we should take it up. I feel my comments may have been very forthright, but that is my job. So I have done that. Thank you very much, Dr. Gupta. And as far as I'm concerned, I thank you once again for the opportunity to be party to this exciting conference. Thank you.
Thank you very much, Brigadier Rajan sir. Thank you very much for your concerns and comments. Now, I request Dr. Ashwani Kumar, scientist from Herbal Division, PRI, for vote of thanks.
Thanks, Prashant sir. Good afternoon, everyone. Firstly, I would like to express my gratitude to Param Pujya Swami Ramdev ji and Param Shraddha Acharya Balkrishna ji for their blessings, guidance, and supervision. A special thanks and appreciation to Professor Bhushan Patwardhan sir, the editor-in-chief of the Journal of Ayurveda and Integrative Medicine, Kavita Narayan Semi ji from LCA team, our renowned speakers, Dr. Manali Joshi, Dr. Saket Ram, Dr. Vedpriya Arya, our dynamic moderator, Dr. Prashant Gupta, our special guest, Dr. Brigadier R.S. Rajan sir, for concluding remarks. Further, I want to thank Dr. Girish Telu sir for his guidance, support, and execution of this event. And thanks are also due to Ms. Sanchita Sangale, the editorial manager of the Journal of Ayurveda and Integrative Medicine, for her coordination and execution. Lastly, I would like to congratulate the organizing team from Patanjali Research Institute, from Journal of Ayurveda and Integrative Medicine, and our national and international participants for making this event a grand success. Now, I request Prashant sir to kindly close the session. Thank you, sir.
Thank you very much, each and everyone. The session is now, the webinar is now over. Thank you all the speakers and the nearly 100 participants. Thank you very much all.